Our brain is the most complex organ in the body. It controls everything. Thought, memory, emotion, touch, motor function, vision, breathing, temperature, hunger, and smell. Smell is the focus of this presentation, specifically the link between Parkinson's disease and smell loss. This link could prove to be a useful tool for diagnosing neurodegenerative diseases like Parkinson's. There are many crucial parts from our nose to the olfactory bulb that allow the brain to process and identify these precise smells. Here's how it works. Airborne particles are released from a source such as freshly brewed coffee. During normal breathing, those particles enter our nose through the nasal cavity. They travel to the roof of the nasal cavity and the lining of the olfactory epithelium. This is where specialized collections of cells, known as olfactory receptor cells, wait for the particles to bind their string-like projections or cilia. Electrical signals are then sent up through the olfactory nerve and the olfactory bulb to the brain, where they can be interpreted as odours. 12 million of these olfactory receptor cells specialize in transmitting a particular odour molecule. Odour molecules attach to smaller protein molecules at the end of each cilia facing the outer epithelial wall. They then make connections that allow electrical signals to travel along the olfactory nerve. Once these signals pass along the olfactory nerve, also known as cranial nerve 1, they reach the olfactory bulb, where axon bundles make synaptic connections with smaller cells that form the olfactory tract. These smaller cells, known as mitral and tufted cells, are in an area called the glomerulus. Humans have from 1,100 to 1,200 glomeruli in the olfactory bulb. Within the olfactory tract, a tube-shaped projection starting from the underside of the medial frontal lobe to the olfactory groove, electrical signals travel to the main structures of the brain associated with smell, as well as memory. The areas associated with smell include the primary olfactory cortex, located on the base of the frontal lobe and inferior surface of the temporal lobe. It is the main site for olfaction and processing odour, and the limbic system associated with memory formation. Many other areas are closely associated with the olfactory cortex, including the piriform cortex, amygdala, and entorhinal cortex. The most misunderstood areas include the olfactory tubercles, the orbitofrontal cortex, and the anterior olfactory cortex, located between the olfactory bulb and olfactory tracts. Researchers are learning more about them. Recent studies suggest that the anterior olfactory cortex is a site where neurons are greatly affected by Parkinson's disease. This is caused by Lewy bodies, clumps within neurons that can contain abnormal misfolded proteins called alpha-synuclein. Alpha-synuclein buildup is harmful to neurons and can easily spread to other neurons within the same area. Alpha-synuclein can also damage other supporting cells of the central nervous system, such as microglia, pericytes, and astrocytes. If the anterior olfactory cortex is destroyed, smell loss could easily occur. Neuron death can also be found in major brain sites affected by Parkinson's disease, such as the substantia nigra. This leads to dopamine reduction, a chemical that plays a vital role in motor movement. That's why people with Parkinson's experience slowness of movement, muscle rigidity, stiffness, and tremors. Alpha-synuclein proteins are the main culprits in all Parkinson's symptoms, but they can also hold key to early diagnosis. Some Parkinson's patients experience motor symptoms a decade after smell loss. That's why it's important not to ignore any smell impairment. It may indicate a serious disease such as Parkinson's.